I was asking, our director of the IAS, Professor Henry Tay, whether he would like to introduce our speaker of today, but he really wanted me to do it. I guess I can't escape, right? So I'd like to introduce today's speaker, Professor Curtis Suttle from University of British Columbia. Professor Suttle is not a stranger to us. He actually has been on our campus a few times. Know our system a little bit, but over the last 2 or 3 days, he got to know our system much better than ever. Professor Curtis Suttle got his bachelor degree in zoology. That's what I really like most. He graduated from the Department of Zoology. That's where I did my Killam postdocs. So in 1978, if I remember all the numbers are correct, okay? And then right after he graduated from the university, he thought, continuous study may be quite boring. And he picked up the technician job in the government, the fisheries and oceans of Canada, for the short period of time. And then he, became the research scientist or something. I cannot remember the titles. But anyways, after a while, he decided to go back to the university. He thought, the bachelor degree cannot really, help him too much. He went back to the UBC and finished his PhD degrees in 1987. Right after he finished his PhD degrees into the, is that Botany department. At that time, they don't have the Department of Earth Science and Oceans Sciences, right? That's just a Botany department. And he moved to New York stock market now. It's a Stony Brook, the New York State University at Stony Brook, and becomes the research scientist over there. He was there for almost about 4 years before he moved to the University of Texas in Austin and becomes the assistant professor there and also research scientist. He was there until 1996. Of course, he was a tenure and became associate professor there in 1996. And then, Vancouver is a very, very attractive city. He decided to go back to his hometown or home universities. So he, was attracted by UBC and returned to the UBC and with a tenured position as associate professor. And then in 1996, that's 1996, and then he became the full professor in year of the 2000. On the same year, he picked up the position of associate dean in the faculty of the science and have been deans until I think the last years. So the Professor Curtis Suttle was trained as a biological oceanographer, and basically he's interested in look at the biological process that controls the nutrient cycling and also energy flow in the marine ecosystems. So most of the interesting topic he really had, still have, is trying to look at the virus. I believe he published his first paper of the marine virus, and also everybody called him the godfather of the marine virus. So we have to keep a distance from him, right? The virus is very dangerous. But if you look at the title of his presentation, it's a quite different story, okay? So they have been working on the marine virus since, and he published substantially in Nature, Science, PNS, and also Eastmill, and lots of the wonderful publications. So because so he has been very, very active, he serves on many ocean science, the journals as editorial board members, also international society members. He wins a lot of the awards for his research in the excellency in the immunology and oceanography. And he was elected as the fellow of the Advanced Institute of the Research of Canada in 2007, and was elected a Royal Society Fellow of the Canada in 2008. I think starting from early 2000, he wins a lot of the research awards of the International Society. The most recent ones, just about a year ago, he wins the Hutchinson's, right? If I remember correctly. The Hutchinson's Research Award for the excellency in ocean science. And year before, he wins a medal, the Tim Parsons Medal, for the excellency in the research in the immunology and oceanography. And year before that, he wins a Hutchinson's Research Prize. So many research prize, research awards. And so he has been, in many places, delivers invited speech. If I remember, his invited talks is about 200 up to this moment. So we are fortunately to convince him to come to USD as the senior visiting fellow of our Advanced Institute of these universities. So we should be able to see him more often in next 5 years, because he agreed to accept the appointment from Henry. That means for the next 5 years, he need to come to USD, at least once or twice the years, So I think, without overdue, I really like to, give this the podium to the Professor Curtis Suttle. So please join me to welcome the Professor Suttle to deliver his speech today.
Thanks so much. This is on, I guess, and thanks for the kind introduction. You know my CV, I think, much better than I do. So, it's a great pleasure to be here. There's certainly lots of seats down the front. You don't have to be shy. So I rarely bite. So I want to talk to you a little bit about some ideas. Maybe, is this too loud, or is it about right? It's okay? Okay. So talk to you about some ideas about viruses, maybe in a way that you haven't really thought about them before. We're all aware of viruses and the fact that they cause disease, that they make us ill. There's been SARS, there's been influenza, there's been lots of things that Hong Kong has been affected by, and in many cases also they come right to Vancouver because we have so many people traveling back and forth. So, but I want to talk to you about viruses in another context. I want to talk to you about how they have another role. And so this title maybe is a little provocative, but I think it's becoming more and more apparent that probably viruses have more of a beneficial role than they do a detrimental role. And so that's the thing I'd like to explore a little bit from the perspective of ocean sciences. And so what we often don't think about is for the first couple of billion years on Earth, there were only bacteria, archaea, and viruses. That was the whole ecosystem. So what we're used to now with all of these other organisms and these complex organisms, in fact, it was much similar. So all the biogeochemical cycles on the planet, all of these different interactions were actually mediated by just these individual players. And So I know everybody here is not a biologist, so just if I spend a moment just to indicate what a virus is. So these are extremely small. I will talk about how small they are in a moment. So they're ultramicroscopic agents. They're obligate parasites. So they, so far at least, although that may change, we've not found any viruses that can replicate on their own outside their host. And they encode their genetic information. They can use, whereas all cellular life forms use DNA, viruses can use, and all cellular life uses double-stranded DNA. Viruses can use double-stranded DNA. They can use negative sense single-stranded DNA, positive sense single-stranded DNA, double-stranded RNA, negative sense RNA, positive sense RNA. So anything you can do with nucleic acids, there are viruses out there that use it for encoding their genetic information. So, so where do we find viruses? So, well, we've looked for viruses in lots of places. We've been up in the Arctic Ocean on icebreakers. I had one of my graduate students from a tropical island on the Indian Ocean. I said, surely you'd like to spend 4 months in the Arctic over Christmas and New Year's, you know, where it's dark 24 hours a day, and all you can do is experiment. So he went up there for 4 months on an icebreaker while it was frozen into the ice and conducted experiments. We've done work in something called the Crystal Caves. They actually have a National Geographic special of when we were down in the Crystal Caves looking for viruses and bacteria there, which is a mine. We've been down 2.5 kilometers deep in nickel gold mines looking for viruses, where the water was last at the surface 10 million years ago, and it's coming out, and we can look at the microbes in there, the viruses. And I've had a PhD student who looked for viruses in hydrothermal vent systems as well. And it turns out that these hydrothermal vent systems are actually, those black smokers, those are actually columns of viruses coming out. It's amazing. They just pump viruses out, and we can follow those columns of viruses for 25 or 30 kilometers. And they're very, very high concentrations of viruses coming out. So what we've kind of got now is this concept of what we call the Virosphere. And the Virosphere is everywhere that viruses exist or are affected by viruses. And essentially, that's everywhere on the planet where you can find life. Whether you go up, I'm doing some collaborative work with a colleague in Spain right now, and we've been calculating the vertical flux of viruses from the atmosphere in a very pristine environment high up in the mountains. It's about 10 million viruses per square meter per day that are coming down, that just from the atmosphere. So viruses are essentially everywhere. And so I want to now kind of move into the oceans a little bit, into water. And let's start looking at viruses in the oceans. And when we started this work, it was kind of interesting because people published papers why there were no viruses in the ocean. And they didn't expect to find any, but it turned out, in fact, that they are incredibly abundant. And so if you were an alien that was to come down onto Earth, and you are not an alien, but you sent a probe, is more likely, you're probably not going to come here, and you were to sample the Earth, this is what you see up there is what it's going to look like, basically, right? The chances of hitting an elephant or any, complex animal is going to be extremely small. You might hit a terrestrial system. You have about a 25% chance of hitting a terrestrial system. So in which case you might find some cellulose or something like that, some complex carbohydrates. But the life forms you're going to find are going to be microbial. Chances are you're going to get some seawater, and you're going to pull it out. And if you had sent many probes, you're going to get water many times. And everything you see is going to be microscopic. So this is a sample in which the nucleic acids have been stained with a fluorescent dye and then looked at under an epifluorescence microscope. And this stain is staining nucleic acids. And so it turns out that this is really what seawater looks like if you were to take a tiny snapshot of seawater. And those little dots that you see there, those are virus particles. And then the next big abundant thing, these slightly bigger, those would be bacteria. And then in the middle there, you'll see one, that's one protist. And so that's what the oceans basically look like. And so So what we know now is that on average, there's about 10 million virus particles in every milliliter of seawater. Okay, so that means that every time you go swimming, you swallow about as many viruses as there are people on the earth. Okay, just from the amount of water that you take into your mouth, right? And it doesn't, and these are infectious viruses, right? But they don't make you sick. And so an anecdotal story, I had the BBC heard about this. They came to my lab and they said, because I think they called me up and said, yeah, you could drink seawater and it's not going to make you sick, so you shouldn't be worried about viruses. So they showed up with their film crew and their beer mug and made me take them out on a boat, and I had to, this 1-pint beer mug kept drinking the seawater because they had to take it from every different angle. And I wasn't really worried, except the water wasn't very salty. And I knew that the sewage treatment plant was around the corner and it was in the fresh water. So, but even then I didn't get sick, right? So most of the viruses out there are not going to make us sick, right? So they're pathogens for other things. So if we start thinking about that, you know, you looked at that image, it's a little bit like an image of, from the Hubble Space Telescope or something. So let's compare it to, say, stars in the universe. So we know that there's about 3 times 10 to the 23 stars in the universe. So this is amazing. In astronomy, you get a paper in Nature by saying we underestimated the number of stars in the universe by 3-fold, right? I mean, gosh, you know, you're certainly not going to, if you underestimate the number of viruses on Earth by 3-fold, you're not going to get a Nature paper, that's for sure. But so it turns out that if you do this, there's about 10 to the 23 stars in the universe. There's about 10 to the 30th virus particles in the ocean, right? So there's a lot more viruses in the ocean than there are stars in the universe. And if we kind of continue that idea about how abundant they are and think about how small they are, viruses are really small. And it's interesting, even though I work on viruses, I just knew they were small, right? I didn't really think about how small they were. And I was giving a talk, after dinner talk, and I thought, well, I better like try and relate to how small they are. And so I said, I was in Toronto, which is where the CN Tower is, which is the largest freestanding, it was at one point the largest freestanding tower in the world, but now it's small, but it's still the biggest in Canada. So, so I said, well, so let me help people visualize it. I'll say, well, if I'm as tall as the CN Tower and I scaled a virus by the same amount, how big would the virus be? So that they could relate to that. So the CN Tower is, so the average, a decent-sized virus is about 100 nanometers in size. The CN Tower is about 325 meters. The average person is about 1.7 meters tall. So it turns out it's about 325 people tall. So if I was as tall as the CN Tower, how big would a virus be? How big would it be would it be as big as a golf ball or something? Or, well, no, I couldn't see it still. So I said, okay, well, geez, that's a surprise. I thought I'd be able to see it if I did it. So let's do the other thing around. I'll make a virus as big as a pinhead. So if I make a virus as big as the head of a pin, how tall would I be? And so if we have a 1.5 millimeter virus-sized particle, I would be 25 kilometers tall, right? So they're incredibly small. And so it's amazing that something that's that small has such a profound effect. And you'd think something that small, so you know, even though they're incredibly abundant, you say, well, maybe that doesn't make a big difference. So I thought, well, okay, so they're really small. So what would happen if we took all the viruses in the oceans and we put them end to end? How far would they go, right? Because they're really small. And if they're that small, probably would they go to the moon? Or, you know, if I took to those 10 to the 30 viruses, you see, this is called displacement behavior. Are there biologists there? I think I was supposed to do something really difficult, like writing a grant. So I said, I don't want to do that, but I can do this in the back of an envelope. This is way more fun than trying to do something that's hard. So I took the 10 to 30 viruses in the ocean. Again, we assume they're 100 nanometers long. So you put them, that's 10 to the 23 meters if you put them end to end. So that's an incredibly large number. So let's divide it by kilometers. That's 10 to the 20th kilometers. Let's just divide by light years. 10 to the 13 kilometers, so that's 10 to the 7th light years. That's how far they would go if you took just the viruses in the ocean and you stretched them end to end. So how far is that? So Proxima Centauri is 4.2 light years away. Crab supernova is 4,000 light years away. The Milky Way is about 150,000 light years across. They would go further than the nearest 60 galaxies, right? If you took all those little virus particles and stretched them end to end. Even though they're that small, right? That's how far they would go. And so that might seem like a really trivial calculation, right? So who cares? But what's amazing, we can start doing other things. It turns out that in the ocean every second, there's about Avogadro's number of infections, 10 to the 24th infections per second, right? And so every time there's an infection, there's an opportunity to laterally transfer nucleic acids from one cell to another, right? You see all of these evidence of lateral transfer of genes. There's opportunities of co-infection. There's you're changing populations. Every one of those 10 to the 30th viral particles has its own evolutionary trajectory, right? So they've got their own genetic history that's tied up in those genomes. So in fact, we're looking at evolution over an amazing amount of time, and it's all archived in that viral DNA, right? So it turns out that so viruses are incredibly small, and there's lots of them, right? And so then what does that mean when we start thinking about those number of infections that are going on? What impact does that have on some of the other processes that we're thinking about? And so that's what I want to explore a little bit. So if we think about those numbers, what does that mean? So if we look at life in the ocean by numbers, if we assume that viruses are life forms, about 90% of the genomes in the ocean are actually tied up in virus particles. And that's just double-stranded DNA viruses. There's arguments there's just as many RNA viruses and just as many single-stranded DNA viruses. But there's a lot of them, right? So about 90% of the genomes are actually in viruses. If you look in weight, that's about 200 megatons of carbon. So if you compare that to blue whales, so in blue whales, the most carbon there was ever in whales was about 13 megatons. That's before whaling, right? And that actually turned out to be quite hard because whales are not usually expressed in units of carbon, right? They're like tons, right? So I had to find a conversion factor to convert tons of whale flesh into units of carbon. So I think I used minke whales or something, but hopefully that's appropriate for blue whales. I'm not exactly sure. But if you look, these are all dwarfed by prokaryotes. So there's about 5.2 gigatons of carbon in the oceans in prokaryotes. So it turns out that if you kind of flip it around, even though viruses are by far the most abundant, they're so small that most of the carbon, in fact, is tied up in unicellular prokaryotes. But life in the oceans, then, is microbial almost entirely. If we look at how that's divided up, so we've got about 90% in prokaryotes, and about 10% in viruses, and about the same amount in proteins. So there's a lot. And so, but why does, so we've got lots of big numbers here. So, and why does anybody care? Well, it turns out that, in fact, that if you take all the living material in the oceans, everything that you can think about, that you would sort of recognize as being macroscopic, whether it's shellfish, seaweed, crustacean zooplankton, whales, anything you want, you could put that on one side of the balance. That would probably represent less than 5% of the living material by weight in the ocean. About 95% of the living material by weight in the oceans is microbial. And those microbes produce about half the oxygen on the Earth. And viruses kill about 20% of that living material every day. Okay, so viruses, so you can start to see then, so you've got something that produces half the oxygen on the planet, also about half the respiration, consumes about half the oxygen, and it turns over that quickly. You can now start to see where viruses now start to have major become major players in global biogeochemical cycles, right? Because they're turning things over so rapidly. You just change the speed at which that wheel turns a little bit, that has large consequences. Okay, so if you're interested in things like global warming or global biogeochemical cycles, you really can't understand that unless you start putting the microbial component into it. And if you want to understand it, you've got to put the viruses into that as well. And so that's a very different role for viruses, right? People didn't really think about viruses as driving global biogeochemical cycles. Okay, so viruses have this other emergent property. So what are viruses doing then? We can kind of think, I want to talk a few minutes to show a little bit of data about viruses. You can kind of think of being catalysts. They kind of lubricate the flow of material from living organisms back into what oceanographers call particulate and dissolved organic matter, so up here, right? So what happens is viruses, they kill phytoplankton and zooplankton and bacteria. They release that material as dissolved organic matter, and that dissolved organic matter then gets consumed by heterotrophic bacteria, and you get respiration going. And so you start seeing you get some pretty significant carbon cycling going on. So that's what we've called the viral shunt. And so if you, again, you kind of take a big picture of this sort of thing. This is quite dated now. But now our fossil fuels are much larger than that now that we do fracking. But there's a huge amount of organic carbon that's tied up in reservoirs of fossil fuels. And if you look at it, we've got, what does that say, 805? So it's probably more like about 830 or 840 gigatons of CO2 in the atmosphere. So I'm looking at where the reservoirs of this kind of carbon might be in the planet. And if you look at, those are dwarfed by the amount of inorganic carbon in the ocean, which is as bicarbonate and CO2, right? So that's about 38 teratons, 39 teratons. So that's massive compared to the amount of carbon that's in the atmosphere. And if you look at fluxes of carbon, we've got about 2 gigatons per year from burning or from deforestation, and about 5 gigatons per year from burning of fossil fuels. So that's about 7 gigatons per year. We get 10 times more just coming out of the ocean, just from gas exchange. We probably got about 90 gigatons per year just degassing from the ocean. But we have about 93 gigatons per year going into the ocean. Okay, so that's a good thing, right? So we're actually decreasing the amount of CO2 buildup in the atmosphere because the oceans are absorbing that CO2, and it's becoming dissolved in the ocean. What's the consequent effect of that? So that's ocean acidification, right? And so people probably are aware of that if you've taken any kind of environmental sciences courses. So the pH in the oceans is gradually dropping, right? And that has large consequence effect for things like coral reefs and calcification of organisms. So we've got this kind of mixed balance. But that reduces the amount of CO2 buildup in the atmosphere. That 3 gigatons per year actually is exported. So this is something, as oceanographers would call the biological pump, which is essentially taking material from the ocean. When it dies, it sinks out, right? So as phytoplankton, zooplankton, fish poop, whatever, it sinks, right? So that's the process which takes CO2 from the surface of the ocean and exports it into the deep ocean. Politicians would like to actually do that more efficiently, because if you can get it below there deep enough, it's down there for about 5,000 to 7,000 years before it comes back to the surface again. It's trapped down there, right? It has other negative effects, like you don't really want the oceans to lose all their oxygen, because that's one of the consequent effects of exporting a lot of organic material. So viruses, so this is what we call the biological pump. So do viruses have some sort of effect on this? So if we look at the biological pump, that's the biological pump, right? It's things, cells and dying and organisms, things like that, sinking from the surface into the deep ocean. So viruses, in fact, will attach to those cells. They will kill them, and they'll release that organic matter, and it'll stay in the surface, right? And it doesn't sink. Okay, and then the bacteria and things will work on the organic material in the ocean. And so the point of this is just to show that viruses change the pathways of nutrient cycling and energy cycling. It's not that they haven't always been doing this. It's just that they have an impact on the system. So rather than cells being exported to the deeper ocean, some of them are going to be recycled in the surface waters. We can think about it, you know, if we kind of illustrate that in another cartoon. So viruses come, they attach to a plankton cell. That cell lysis, releases dissolved organic matter, like we saw, in particular organic matter. That gets attacked by enzymes and things like that. Ultraviolet radiation breaks it down. That material then is released back into the dissolved organic matter pool, taken back up by the bacteria plankton, and respired as say, CO2, right? So you've got all of this kind of complex carbon cycle. We're just talking about carbon, okay? So there's effects, right? So do viruses then, so you can see that they're starting affecting the mobilization of carbon. So what other kinds of effects do they have on plankton communities, right? So they can affect, they're obviously killing organisms and they're releasing that material. Do they have other kinds of effects? Do they have positive effects, okay? So we did some experiments. We were out on a ship in the ocean. And we came across a big bloom. So a bloom is where you have a really large amount of biomass of one kind of organism. And it was something called Synechococcus, which is a cyanobacterium. And it's one of the most important primary producers in the oceans. Cyanobacteria is pretty famous, right? Because they invented oxygenic photosynthesis about, I don't know, 2.5 billion years ago or so, and that killed just about everything on the planet because oxygen is terrible stuff, right? Because it oxidizes everything, right? So all those poor anaerobes that, you know, where are they now? They're in hydrothermal vents and hot springs, right? That's the only place left where they can survive, right? And in your gut. But there's not many places left for them, and so because oxygen took over, and cyanobacteria were the guys that did that for us. And there's a bunch of viruses which infect cyanobacteria, especially Synechococcus, which we had been able to isolate. So we came across this big bloom in the ocean. We said, here's the perfect opportunity to see what is the impact of viruses on mortality of cyanobacteria. So we set up these experiments to look at that. And so you see these two guys here, they're smiling, right? Because those were my first two postdocs, Steve Wilhelm and Marcus Weinbauer. And it took us 22 years to publish this paper. So by the time we published this paper, Steve Wilhelm was a full professor, and Marcus Weinbauer is a senior scientist at CNRS in charge of a whole program in France, right? And why did it take us 22 years to publish this paper? Because it didn't I keep telling my students it could get worse, right? You know, it's only been a couple of years, right? So we've got to get this right. And the reason that I wouldn't publish it, because I didn't understand what was going on. And what was happening is, so what we were doing is we were changing the concentration of viruses in the ocean we could do that by using some techniques. And as we took the viruses out, we were expecting to see mortality decrease and growth rates increase. It was exactly the opposite. So the lower the concentration of viruses, the slower the growth rate. And then we took all the viruses out, they just stopped growing altogether. There was just no growth. So there was no primary production when we took the viruses out. And as we added more viruses back, things grew faster and faster. And it was directly proportional to the concentration of viruses. And so we're saying, well, this is exactly the opposite of what it should be. As we add more viruses, more things should die. So we should see more mortality, but we're seeing more growth. And so this experimental system here is, so what you can see on the y-axis is something called frequency of dividing Synechococcus. And it's a pretty simple concept. So Synechococcus is a unicell. If it's growing, it's dividing. And so there's a relationship between the percentage of cells that are dividing and how fast they're growing. The faster the population is growing, the more cells are in division. So there's a relationship between frequency of dividing cells, or proportion of dividing cells, and how fast things are growing. So that's the y-axis. And then on the x-axis, we have percent dilution. And so that's the, in terms of how much things are diluted out, so either with high concentration of viruses or low concentration of viruses. And so you can see when we have high concentration of viruses, the fraction of dividing Synechococcus is much higher than when we remove them. And so, as you can see, that goes up. And so we did that in the Gulf of Mexico. I went on sabbatical, 10 years later in Marcus Weinbauer's lab in France, and we did a whole bunch more experiments in the Mediterranean. Every experiment we did, we saw the same thing with viruses, without viruses. Colleagues started telling me they were seeing the same things in lakes, and so it was pretty clear that viruses were stimulating the growth of the primary producers, not inhibiting it like we thought. I had a PhD student then kind of look at this a little bit more. So Emma Shelford here and Matthias Mittelbo, who's a colleague that came over on sabbatical into my lab. And so what you see on, again, the y-axis is chlorophyll, which is a proxy for the phytoplankton because they're plants. So the more chlorophyll you have, the more plants you have. And so we incubated for 12 hours, and we looked at how the chlorophyll concentration changed, again, with viruses and without viruses. And we had data from the Indian Ocean, which is might be similar to offshore in Hong Kong here, very low nutrients. And then we had more highly productive waters. False Creek, in fact, is the ocean, but it's a little embayment near Vancouver. And again, with viruses, we found much more chlorophyll, much higher growth rate. So again, the same sort of thing was going on. So what was happening? So what was interesting is when Emma started looking at the nutrient concentration, she found in the presence of viruses, we had much higher concentrations of ammonium. And so why is ammonium important? Well, phytoplankton, they're primary producers. They're plants. So they don't have, we're heterotrophs, right? So we have to eat hamburgers or I don't know, hagao or something, so that's my favorite anyway. So we have to eat things because we're heterotrophs. We have to take in organic carbon. Plants, they've got us beat there, right? They just make their own carbon. They just need some air and water and sunlight, and away they go, but then they run out of other things. And in the ocean, one of the things they run out of is nitrogen. And ammonium is a byproduct of zooplankton excretion. Lots of things release ammonium. That's what fuels the primary production. And so here we're sitting in a situation where we see in the presence of viruses, we get lots more ammonium production. The phytoplankton are short of nitrogen. Ammonium is a great nitrogen source. So that's what's fueling the primary production. Where's the ammonium coming from, right? Viruses are not metabolically active. They can't be making ammonium by themselves. So what's going on in this system in which the viruses are releasing ammonium into the system? Well, it turns out what's happening is that the viruses, in fact, they're infecting bacteria, which is kind of what we would expect, right? So when they infect bacteria, here's the viral shunt, right? They're releasing this dissolved organic matter, in particulate organic matter. That's going to be kind of in the same elemental composition as the bacteria are, right? So bacteria, heterotrophic, us heterotrophs, we have about 5 atoms of carbon for every atom of nitrogen. Some are 4 to 5 to 1, something like that. Okay, so then when you lyse a bacterium, that's the same thing that's going to happen. You're going to produce 4 or 5 atoms of carbon for every atom of nitrogen. But then there's bacteria there that are not going to be infected by the viruses, because viruses are very specific in terms of what they infect, right? So they're going to infect just certain bacteria. So they infect those bacteria, and the bacteria then take that in. They're heterotrophs now. We're talking about heterotrophic bacteria. They're taking in the carbon and nitrogen, but they're heterotrophs. So what do heterotrophs have to do? We have to respire, right? So if we have 4 to 5 to 1 atoms of carbon for every atom of nitrogen, we're going to have to take in more than 4 and 5 atoms of carbon because we're respiring carbon all the time. Fortunately for us, we just about respire all the carbon we take in. Otherwise, we're all going to look like giant sumo people, right? So mostly, our growth efficiency is incredibly inefficient at this point. So a bacterium has got that same issue. They're taking, in order to grow, they have to take in enough carbon to satisfy their respiratory needs, their metabolic needs, and they have to have enough carbon to make more biomass. So they actually need carbon more like 8 to 1. They need like 8 atoms of carbon or 7 atoms of carbon for every atom of nitrogen. Does that make sense? So what they're getting supplied with, though, is 4 or 5 atoms of carbon for every atom of nitrogen. They don't have enough. So what do? They deaminate the nitrogen because they can't use it, right? They've got too much nitrogen. They just get rid of it, and they spit it out. And that fuels the primary production. So it turns out that viruses, in fact, are liberating nutrients, which is fueling the primary producers, which allows them to do more photosynthesis and put more carbon into the system. So again, something that's not what we would have necessarily intuitively thought. So we're involved in a project right now with a group of scientists from MIT and Princeton. And we're actually trying to put this into a global context and look at what proportion of the carbon which is produced by primary producers are in fact lysed by viruses. And so what we can see then is we've got, and we're still, the numbers aren't quite right yet, but we're trying to put this model together so we can start making predictions. Okay, so the point of this is that viruses influence global biogeochemical cycles, right? So that's what they've been doing for billions of years. Remember, they were the only predators in the system. We didn't have any other predators, right? They were just viruses. They were photoautotrophs, they were heterotrophs, they were chemolithotrophs, right? All these things that would grow. What got rid of the biomass? Viral lysis. Okay, so that's what would liberate the organic nutrients. So then another obvious thing is, well, we've got lots of viruses out there. Pretty obvious next question is, what are they? And so I'd like to spend the rest of my talk kind of thinking about what do these viruses mean in terms of life on Earth. And so if we start thinking about this is, how do we look at that? We look at, how many here are like biologists or have biology backgrounds? Most people? Okay. Some not. So I've tried to give some back. But so if we look at, I'll give a little bit of information as we go along. But if we look at genetic diversity, we look at that by typically looking at nucleic acids, right? So it's pretty intuitive. If we were all identical, our genetic, if we're all exact clones of each other, we'd have the exact genetic information, right? Our genetic diversity would be 0, right? We would be identical. And we'd look at that by looking at our genetic information. So if we start thinking about that, and we start thinking about that in the context of biodiversity, my lab is in a biodiversity research institute. And my colleagues would say, you're not really working on biodiversity, you're working on genetic diversity. And I go, oh. Because they work on things like lions in the Serengeti and bromeliads in tropical rainforests and fish in the Arctic Ocean, and they're really working on biodiversity, polar bears, all this sort of thing. But I just like work on viruses, so I'm not really working on biodiversity. So I sort of thought about this for a while, and I said, well, so I gave a talk, and I said, well, I've got to stake my claim that I in fact belong in this institute at all. And so I like the definition that the UN has. And the UN came up with this idea that biodiversity is the totality of genes, species, and ecosystems in a region. And so I thought, well, that resonates kind of true. And I think if we think about that intuitively, what that means is what underpins all biological diversity is genetic diversity. Just like we said, if there's no genetic diversity, we have no biological diversity. So if we make that assumption, then the more genetic diversity we have, then that's the more biological diversity we have. That's the more ecosystem diversity we have, right? And so then what's really critical is, where does most of that genetic diversity lie? Is going to be really critical in terms of trying to figure out where most of the biodiversity is. So the point being, then, the roots of biological diversity and ecosystem function then have to reside in the overall genetic diversity. Okay, and so we think about that, and in particular for those that don't think about these questions very much, if we start looking at kind of the big tree of life, what we call the big tree of life, we use a gene for that. We use something called the ribosomal gene, because all living organisms have all cellular life have ribosomes. And so we can look at those ribosomes. And if we do that, we can kind of divide the tree of life up into 3 major limbs. One are the bacteria, right? Another, which people didn't realize were so different, but they really are something called archaea. They're as different from bacteria, and archaea are very different. And then the third are the eukaryotes, or the protists. And you can look at those, and you see those are the, and then people might look at this and say, well, okay, but where am I on this tree? And so, well, there's the mushroom way out there, right? And I'm right there, okay? So maybe your closest relative isn't a mushroom, but my closest relative is a mushroom, right? So the point is that we're almost indistinguishable from mushrooms, at least in terms of genetic content, right? Everything from, there's corn. Everything from here down is microscopic, right? And the distance on the tree is how evolutionarily distant you are, right? And so essentially all us metazoan things, from corn to mushrooms to humans, are right at the tip of the branch of evolution, right? So, and fish would be right there as well, right? We probably couldn't distinguish me from a fish hardly. So the point being then, if we look at this big tree of life, all the diversity essentially is microbial. And what if we throw viruses into that tree of life? Where do the viruses show up? Well, the viruses are so distant, we can't even put them on the tree of life, right? They have their own flying saucer through space of diversity, right? So they've divided it up into single-stranded DNA, positive sense, negative sense RNA, these sorts of things, right? But they are so divergent from cellular life that we don't recognize it. And so if we want to start looking at diversity, we look at genetic diversity. And so most of your biologists are familiar with it. We use the genetic code to kind of unravel diversity. And so we have these GCAT base pairs, which ultimately translate into amino acids, which make proteins. And the point that I want to make is that proteins are critical, right? Proteins are those are the functional units of life, right? So, I mean, we have structural proteins and things like that as well, but all these metabolic, catabolic processes and things are actually driven by proteins. So it's that sequence. We can look at those sequences then, and depending on what we know, we can infer what the function of certain proteins are, and we can also make inferences about diversity. And so what do we do when we start looking at these questions? Well, we go out in ships, and we collect a whole bunch of water, and then we take the viruses out of that water. So we might collect about 200 liters of water or something like that. We'll pass that through a series of filters to remove the big things, like proteins. Concentrate the viruses and the bacteria, and then we'll concentrate the viruses, we'll extract the nucleic acids, and then we can start interrogating those and asking questions about what they are. And so you get that genetic information. What you typically do, we'll take that, look at that, all that nucleic acids that you get, and then you'll compare it with known genetic information. And then you'll, based on that comparative information, try and figure out what's going on. And so we can do this now a bunch of sorts of ways. We can use something called metagenomics, which is where we take all of the nucleic acids, say from the water, extract it all, chop it up into little pieces, and sequence it randomly. We can do something called phosmid cloning. And I'm going to touch on each of these very briefly, where we take a bunch of DNA and put it in a big piece, so then we get all that genetic information in a genetic context, in a genetic area. We can do gene surveys where we pick something this is like that ribosomal tree. We pick a gene that we know really well, and we sequence it deeply. Or we can take new isolates, and then we can try and see what's related to this particular thing. So the first time we did this is in a collaboration with a fellow called Forrest Rower's group at San Diego State University. And we've been collecting this virus stuff for a long time. We have more than 3,000 virus communities that we've collected all over the world, and we've archived them. And so I started doing this in the 80s, thinking, and that's when we were sequencing on slab gels, right? And I thought, eventually the technology is going to catch up where I can actually figure out what's in these samples. But at that time, you got a PhD for sequencing a gene, right? So, and that would take 4 years to sequence a gene, right? And so there was no way that we had the ability to do it, but I started saving all the samples, right? So we just started saving them everywhere we went, from the Antarctic to the Arctic, all over the world. So I have this huge library. And so Forrest and I sat down and said, well, why don't we just take that And let's just start asking a big question. We'll take a bunch of samples from the Arctic Ocean. So I took 56 virus communities collected over a year in the Arctic Ocean. We took 85 communities collected over 8 years in the Pacific, and 41 communities collected over 5 years in the Gulf of Mexico, and we had 1 from the Atlantic Ocean. And we said the idea was we'd collect all of these things, and by collecting so many samples, we would smooth out, variation. So we'd say and if we could still see differences, say, between the Arctic and the Pacific, then the differences are probably real because we've combined so many samples together. And I'm not going to go into, it's really an interesting story, but the thing that really kind of blew us away when we looked at the data is that more than 90% of the sequences that we pulled out of the virus had no matches in any database. So they had they had nothing that had any recognizable similarity. And so you think about this, and if we think about bacteria, people talk about, oh, the bacterial diversity is high, right? You've got all this amazing bacterial diversity. If you go out in the ocean and did the same thing, about 95 to 96% of the sequences would have matches in databases. Okay? Just about all of them. And it doesn't mean you know what the function of all of those are, but they have matches. They've been seen before. We're seeing 90% of the sequences that have never been seen before. And the other thing, when you look at bacteria and you find sequences, they're usually prophage that are in the bacterial genomes, right? And that's why they haven't been seen before. So from this, we realized that the diversity of viruses was absolutely enormous. I had a PhD student, a postdoc in my lab, Alex Cully and Andrew Lang. We did the same thing with RNA viruses, the same story. About 80% of the sequences had no matches in any database for RNA viruses. And the ones that did have matches were from just about all the different families of viruses. They were very, very different. And then we looked at single-stranded DNA viruses. So when we did this experiment with Forrest Rower, one of the surprising things is we came up with a lot of sequences of single-stranded DNA viruses. And people didn't really know there were single-stranded DNA viruses in the ocean. So we thought, okay, so Jessica Labonte, a PhD, so why don't we go out there and see what's in the single-stranded DNA viruses? And the neat thing about single-stranded DNA, if you find it, it's viral. Nothing else makes single-stranded DNA. So she went out and purified the single-stranded DNA. And for those of you that are interested in research, this is what happens, right? So she came into my office, and she's practically crying, right? She's just so upset. And I said, well, why are you so upset? And she said, well, I got all my data back. And I said, well, great. Well, what is it? She said, I don't have a hit to anything. And I said, well, you rock, because you've got single-stranded DNA. Because if it was anything else, you'd have a hit to something, right? And so she had all of this data, which had never, basically sequences that had never been seen before anywhere. And so she took that data, and so it's always a problem. What do you do with this if you have nothing to compare it to, right? What do you do with that information? And so what Jess did when she collected these samples, so when she looked at it hard, she realized, okay, about 10% of the sequences actually do hit stuff. And those 10% of the sequences hit all the known families of single-stranded DNA viruses. One's infecting bacteria, one's infecting plants, one's infecting animals, right? So she covered everything that was known. 90% didn't hit anything. But she was able to assemble that, and she assembled assembly means stitching the genes together. And so she ended up with about 650 complete genomes. And then when she started looking at those, she came up, so there's the families of single-strand. There's 7 families of single-stranded DNA viruses that are known or recognized. So when she looked at her data, I don't know if I have it on here or not. So, so I've got it. Yeah, so when she looked at her data, she was able to do some sophisticated analysis. She ended up with about 125 potential new families of single-stranded DNA viruses, right, from a few water samples. So again, it just shows this enormous diversity. All of those viruses are out there infecting organisms, and we don't know what any of them are doing. Okay, so it's amazing. And then when she was able to do that, we could go back to these other data sets, and we could find a home for about 75% of all the sequences that we pulled out would fit into one of those 125 evolutionary groups. Okay, so we were able to do that. Another approach we can do is trying to do phosmid cloning, and this is Danielle Winget and Cheryl Chow, postdocs in my lab, had worked on this. And the idea was, because, well, we have all these little pieces of DNA. It's driving us crazy because we have, so what the equivalent of this is when you think about metagenomics, you have, let's say you have a jigsaw puzzle for every organism that you have out there, right? Like how many genes are there? So megabase of DNA or something's in a bacterium, right? So we've got, say, I don't know, we've got 1 to 2,000 genes. Okay, so that's what you've got, about 1 to 2,000 genes per bacterium. So let's say each one of those is a jigsaw puzzle. A gene is about 1,000 base pairs long or something like that for a small one, something like that. So we take those. So each one of these, this is their genomes. We've got, say, 1,000 different kinds of bacteria out there. We take those puzzles, we throw them in, one for every bacterium, and we break them, and we mix them all up. We've got 1,000 different puzzles now, right? We've got them all there. And then we do metagenomics. So we go in there with a bucket, and we take a bucket out. And then we throw them over there and say, OK, let's put them all back together again. But you've only got like 2% of the puzzles, and you're trying to figure out what everything is. And it's cut up into like each piece is about less, maybe 5% or 10% of a gene, right? And so you're trying to stitch all those things together. That's what we're trying to do with metagenomics. So the thing is, if we can get that, rather than having them into little pieces like that, if we can get them into one big piece, then we've got a genomic context, right? And so that you can take all those little pieces and say, oh, it fits in here. So you've got a puzzle, you've got a template, so you've got puzzles that are mostly built. And then you can find a piece and say, oh, it's just like that piece, it fits in there. So you can, and that's the idea with phosmids. And if we do that, what you see here is you can put all little bits and pieces in different parts of the gene. So this is where we've been able to recruit metagenomic data onto these phosmids. And so you start to see some really, really interesting patterns then, and trying to close this genetic space. And so we can do that from metagenomic data from the Arctic Ocean or stromatolites or from some of our samples from British Columbia. You can start finding genomic homes for those. And when we start doing that, it starts to be really interesting. So gray in this bar, each one of these lines represents a different phosmid. The gray represents sequences that have no hits to anything. And the different colors represents things that have hits to other things. And in some cases, we've been really lucky. We found viruses that have perfect hits to cellular life, which means that virus is infecting that cell. And we can start putting this stuff together. So that's one way of doing it. The phosmids. The other thing, we can do gene surveys. So we can pick a gene which is representative of a certain group of viruses. And we can ask, well, if we take a very small group of viruses and we pick a gene, how many different types do we have? So here I have 2 examples. One is something which is related to T4, which is one of the most famous viruses infecting bacteria, and one that infects eukaryotic algae. So you can take those. We can look at those. This is Alvin and Danielle. And then you can say, we can take those and we can put them into evolutionary groups based on their sequence identity. And so if you do that, you get all of these different evolutionary groups. We can look at each of those samples and we can say, this is a frequency distribution here, so the number of different evolutionary groups versus the number of sequences that we collect. And if they flatten out, it means we're sequencing enough, right? Because no matter if we take more sequences, we don't get any more evolutionary groups, right? So it flattens out. That's good. That means we have enough sequence. But for every sequence, the pink is the total. And so what you see is that the pink keeps going up. So that means that every time we take a new sample, most of the viruses have never been seen before. It just adds. It keeps going. And so we don't know how. We haven't got to the point now where we add more things that we don't know how many more samples that we've got. OK? And so we can look at those. And so this is the same thing. It shows us the number of different evolutionary groups versus these. So the number within an evolutionary group, the number of different evolutionary groups. Each of these represents a different sample. And you can see there's very little overlap. So each sample is quite different. So we've done that. Is another way we try and tease apart these sorts of questions. OK? And then the final thing I think that we can do, which is really important, is we can isolate new kinds of viruses. And we can go back to the metagenomic data and ask, what do those viruses represent? And again, so here's a new evolutionary group of viruses that we've isolated. and we look at metagenomic sequences that we can recruit all around the world, and we can see how abundant is that new group of viruses in the ocean. We can start recruiting all those little pieces. We can start putting them together, and then we can start closing the sequence space. We can find homes for all those little puzzle pieces, right? We can say they all get together. And so this is what people are trying to do now to try and figure out what does all that diversity represent. So this one in particular was quite interesting because this was a virus we isolated from a meltwater pond on an Arctic ice shelf, which infects a cyanobacterium. When we looked at the sequence information, it turns out that these viruses are incredibly abundant in the Mediterranean Sea, right? So what we're finding is groups of viruses, in fact, probably we don't know existed at all, and now we're trying to look at we can look at their global distribution. And the final example I want to talk about is a really cool one because it shows how we can learn about what these viruses can tell us about evolutionary things and how they can really inform us. So we work on like the smallest viruses are single-strand DNA viruses. They're tiny. They're like 2 genes, 2,500 base pairs. At the other hand, we work on things called mini-viruses, which are like a megabase in size, 1 million base pairs in size, right? And so one of the giant viruses we isolate infects microzooplankton. And it turned out to be really interesting. And so we started looking at it. Matthias Fischer, who was a PhD student, started looking at this virus, sequenced it. Turned out to be one of the most complex viruses that had ever been found. It has all kinds of different genes in it for different things. But what's the real cool story is we had isolated this virus like 15 years ago. When I was still, more than that, 20 years ago, we'd isolated this virus. I had like 4 or 5 PhD students and a couple postdocs work on this, and they'd go and they'd just like, it's too hard, can't do it. They just like, put it aside. And the reason why was because, and when Matthias started looking at this thing, he realized that when he looked at it carefully, so you would look at, here's the little microzooplankton, and it has something called the virus factor. It produces these really giant, big virus particles, which kills the zooplankton. But then there's all these little tiny viruses. And these little tiny viruses actually replicate on the big viruses. They kill the big viruses, and they rescue the zooplankton. The zooplankton survives. And it turns out the zooplankton actually transports in these little viruses. It goes out and finds them and brings them in, because they kill the big virus. And so it's like an adaptive immune system, if you like, right? It actually goes out and finds these little viruses. And so when we went, and so this is why all my students gave up, is because what would happen is this little virus would start to grow up, and they could never replicate the big virus. It would start off, everything would be going great, and then all of a sudden, I can't kill this. I can't kill it anymore. I can't kill the zooplankton. It's like become resistant. But it hadn't become resistant. This little virus would grow up, replicate on the big virus. And then when we sequenced the little virus, it turned out not to be related to other viruses at all. It turned out to be related to transposable elements that are found in higher eukaryotes. And so, in fact, so what happened over evolutionary time is the eukaryotes said, it's a lot of trouble to go out there and find these viruses and transport them in, and what's the chance of actually being successful with that? So it actually incorporated the genetic information from these little viruses into its own genome, and then it became resistant to infection by the big viruses. And then the big viruses died off, and these became transposable elements. They just became jumping genes, right? Because it has no function anymore. There's no selection for them anymore because there's... And so Matthias now has gone off, and he's a professor at Max Planck, and he has now been able to get the little virus to jump into the genome of the zooplankton and actually show that it confers resistance. So he's actually been able to move it in. And so here's something where a virus from a little zooplankton in the ocean tells us something about the evolution of higher eukaryotes on land. And this is just a schematic of that, so I won't go through that. So just to kind of wrap up here. So I think the point here is that I want to emphasize is that most of the genetic diversity on Earth, so in essence, most of the biological diversity, actually resides in viruses. Okay, so if we're actually thinking about biological diversity, we have to think it in the context of viruses. And I think what's really cool, The Economist picked up on actually the story of the giant virus, and I didn't know anything about it at all, and it just happened to be at the same time the Nagoya conference was going on, and Nagoya Conference for Biodiversity in Japan. And what they said, which I thought was very poignant and something that I wish I had thought of, they just said, biodiversity isn't all about, the whales and butterflies and trees and tigers and coral reefs and tuna and things like that. We have to think about it in the context of all these tiny creatures that are too numerous to count and are too small to see, because that's where most of the biodiversity resides. And just to think about this in another context, that one of my PhD students, Caroline Chenard, her research site was at 83 degrees north, right? So that's where people go when they try and go to the North Pole, right? They go up there, and that's as far north as you can go on land, basically. And right, the northern tip of Ellesmere Island in Canada, and you try and get to the North Pole, and you may or may not make it, right? So that's where her research site was. And if you look at these environments, they're intriguing, because what you have are these giant, it's just like British Columbia or Norway, you have these giant fjords that come out from the land. And what those fjords are big valleys that have been cut by glaciers. Okay, so they chop away all the rock, and you get these long, long inlets out there. At the Arctic Ocean, at the edge, there's what we call the ice shelf. And essentially, it's a giant ice dam. It goes down maybe 10 meters, 15 meters, 20 meters, even deeper, where the ice is piled up at the front. And fresh water floats on salt water. It's less dense, right? So what happens is, as the meltwater comes off of the glaciers, it backs up behind these giant ice shelves. And it's been backing up there for 30,000 years. So those freshwater lakes have been there for 30,000 years. They're interesting. They're freshwater lakes with a saltwater bottom. And if we look at the viruses in those systems, it's really intriguing. They're completely different than the viruses in the Arctic Ocean. You might think they might be related. When we looked at the viruses infecting cyanobacteria, where were their closest relatives? Viruses in rice paddies in Thailand, right? They were completely unrelated to the viruses that we would find in the Arctic Ocean. What's going on? So if you look at Carolyn's field site there, so she was working, here's the Markham ice shelf, and you can kind of see it. In a period of about 7 days, that ice shelf broke down completely, and the whole fjord just drained, the freshwater lake drained out, and the seawater just flooded in. And it was gone. And so we've done a little bit of sampling just to know that those fjord microbial communities are completely different than anything else we've ever seen on Earth. And yet, now we've lost all of that diversity without ever really knowing what was there. And so I think it's going to be a while before Greenpeace is jumping up and down saying, save the viruses, save the viruses. But if you actually think it in the context of biodiversity, that's where most of the genetic information resides that we know absolutely nothing about, right? It's really intriguing to me still when we think of the millions and millions and millions of proteins that we've seen now that have no matches to anything in any database, right? So all of those proteins are coding for functions, and we have no idea what they're doing at all. I mean, one great example is a fellow, a bioinformaticist in Switzerland. He went and looked at these metagenomic data sets. And if you think about network analysis, if you haven't, so there's lots of kinds of network analysis, but one simple idea of network analysis is if an organism does photosynthesis, and you were to look at all the metagenomic data and you found, here's a protein for PSBD in chlorophyll, part of the reaction center for chlorophyll, right? So you pull out that protein, you'd start saying, I bet I'm going to find PSBA, PSBE, you know, all of these other proteins involved in photosynthesis, right? You'd bet your house on it, because that's the only way those proteins are all associated with the photosynthetic pathway. Same with respiration. Find a couple of proteins, you could bet your house that you're going to find lots of other proteins involved with respiration. You're not necessarily going to find respiration and photosynthesis or you know, some anaerobic metabolism all at the same time. But if you find one of those proteins, you can build a network. They're highly networked. You find one protein, you'll always find the other proteins, right? Does that make sense? So he went in and looked at these metagenomic data sets, found clusters of highly networked proteins, not one of which was known. So that's telling us there's whole biochemical pathways out there which are doing who knows what, and we have yet to discover what they're doing, right? And so I think that's, it's a difficult problem. We still don't have ways to screen for things that we don't know what they do, right? We can get them. People have done this. You can make beautiful proteins. They can clone them. They can make beautiful proteins. They look at them and they go, do something. So, you know, they drop them in solutions and see if they go blue, right? So that might work if it's an ATPase or something like that. But for most of the things, we have no idea what they're catalyzing. We have no idea what to look for, right? So, so just to end up, I would say, so viruses as guardians of the planet, well, I just think they're the most abundant life on the planet, right? In terms of life form, they're by far the most abundant life on the planet. They affect pathways of nutrient energy flow. They're absolutely crucial to the planet functioning. They probably played that role for most of evolutionary time. I have this theory that I think they archive most of the genetic diversity on Earth. So the interesting thing is people used to think, oh, viruses are escaped nucleic acids from cellular life. Just from the data that I've showed you, you cannot build viruses from cellular life because none of those genes have ever been seen before. But if you look at viruses, they're full of cellular genes, right? They've co-opted, there's whole groups of, so viruses that infect, they encode photosynthesis. There's a whole thing of viral photosynthesis where they infect a cell, host photosynthesis shuts down, viral photosynthesis spins up, right? Those are genes they've co-opted from cellular life. If you look at it, you find cellular genes all the time that have been laterally transferred into viruses. So I think they probably archive pretty much all of the genetic diversity on Earth is archived in viral genomes. Cellular life does not archive viral genes for the most part, okay? And yet most of this is unknown still. And finally, I think we actually risk at losing most of this genetic diversity without ever knowing what it is. So thank you, and bye.
Thanks, first of all, for this exciting lecture, and I'm happy I was replacing my own boring lecture by yours. At the same time, my students are still here, excited. I have a philosophical question, maybe, after all these miraculous things. Maybe you know your neighbors in the US, 25% believe in intelligent design.
Actually, I think it's more like 50%.
What would you tell? I'm not sure. Canadians, maybe not so many. They're more educated. What argument would you bring if you see all these things? Can it be some intelligent design things because you don't have precursors, you don't find it now anymore in the evolution? It just sounds like it's so smart.
That's an interesting question that I would say one that I haven't really, that I haven't, so could all of this, I guess you're just saying, could have all this just be created? But if you look at the proteins, you can actually draw large evolutionary trajectories from them. So I would argue strongly against it in the sense when you look at, say, the photosynthesis genes in viruses infecting cyanobacteria, you can see the one point that they jumped into viruses, and then they radiated tremendously. And so now we have viral it's clearly viral photosynthesis now, but you can see the one point in evolutionary time that it jumped in and radiated throughout all these viruses. And you can go back and you can look at, say, the viruses infecting cyanobacteria are the same 3 families of viruses which infect all bacteria. And so you can say that those viruses probably predated the splitting of the cyanobacteria from the rest of the other bacteria. So, they do make evolutionary sense. We can find these proteins, and you can do beautiful phylogenetic trees. It just, we don't have any idea what they're doing, right? So they're not just random. Yeah, I mean, people ask that question, is because viruses just mutate. They don't actually, especially double-strand DNA viruses, they have the same DNA polymerase as we do, so they have proofreading and error correction and all those sorts of things. So they're actually pretty good at replicating themselves conservatively. RNA virus, single-strand DNA virus, not so much. But you get nice evolutionary trajectories. I mean, so there's nothing that's inconsistent with evolution. Yeah.
I was asking, what about the proteins? So you finally got there. But then I love this idea that you can find networks of proteins that have no known function. So that raises the question, since the viruses are obligate parasites, could these be networks of regulatory elements that co-opt entire systems of a cell's enzymes? And what you really should be looking for would be docking sites on existing proteins based on the structure and how it might fold.
Yeah, I mean, so I only told part of the story there. So when I talked about the network story, what I didn't say was that these were actually networks of bacterial proteins, right? So, and again, these are proteins that have been seen before. So everybody's seen these proteins before. Nobody knew what they do, and now that they find them in these networks, they're obviously, so nobody's done the same sort of analysis with viruses. But what they have done is what they're doing, which I think is, maybe it's similar to that. What we're doing is viruses have distributed metabolism is the idea that's going on, because they don't encode huge amounts of genetic information. But if you look at each virus, which infects a group of organisms, you'll find all of these. You'll have a core set of genes which would sort of define that virus. And I have big arguments with my colleagues because when you look at evolution, how do you look at it? So if you look at viruses, say infecting eukaryotic phytoplankton, or you'll find maybe 20 what I'd call core genes. So they all have these core genes. They also have another 700 genes which are kind of random, right? And so I always say, well, if you give me those core genes, I can tell you all about that virus. I can tell you what kind of organism it affects, what this and that, what group it's in. And my colleagues say, well, you can't talk about evolutionary history of that organism if you're only choosing, hand-picking your 20 genes out of 700, right? But I think the rest of those genes are just, whether you eat with a knife and fork, whether you eat with chopsticks, whether you like to wear a red sweater or a blue sweater, those are just things that are picked up which allow it to do different things, but are not core to its evolutionary trajectory. And so when you go back and look at these genes then, within a group of viruses, you have the core genes, then you have this massive pangenome, which I call it, which is just kind of shared amongst all the viruses. So you start putting like biochemical networks together, looking at the viral genes, you can start building all these biochemical pathways from the viral DNA. But it's distributed amongst all the viruses. So I don't really know what that means. It seems like that there's advantages and disadvantages for carrying all of these sorts of genes. And so there's obviously some evolutionary advantage to carrying them over time, but all of them don't have them, right? Unlike the photosynthesis genes, there's such strong selection for them, just about all of them have the photosynthesis genes. I don't know, that probably doesn't answer your question at all, but how about some student questions?
Thank you. I have one question is, because now it's global warming and the glacier is melting, and because from your talk, the virus in the ocean is adapt, or they are shaping today's ecosystem, and those viruses or microbes in the glacier ice, they are for ancient ages for their ecosystem. So I wonder, what would be your views on where the old days, the ancient viruses are coming back into today's ecosystem? Because from epidemiology, we know that some viruses which kills a lot of humans in 80s, but after 100 years, they just vanished, and after 100 years, and human became vulnerable to those viruses. So would you expect the ecosystem to be changed, or what do we expect for those ancient viruses to come back to today's world?
So we have looked in ancient ice. We've gone back 100,000 years or so looking at ice cores for old viruses. And people have done it in permafrost as well. And you can find them. But they're pretty much, I mean, 100,000 years is just a blink of the eye, right, in terms of viruses. And I think just about all the diversity was already there. So they look a lot like the viruses that we see now. And I mean, it's true. I mean, I think you could probably find pathogens if you went somewhere and you started digging up smallpox victims from ice or something like that. But these viruses are so specific, you'd really have to go out there and find viruses infecting humans if they're going to have an impact on us. In terms of the ecosystem, I think these viruses are just circulating all the time. There's so many of them that I don't think they pose any risk, if that's your question. I think that we're continually seeding viruses. We've done some work, for example, where we can go into sediment cores in the ocean. We can pull out viruses which are at least 100 years old that are still infectious, right? So, and they've been buried there in the sediment. And they can be resuspended and reintroduced into the environment. So there's certainly some sort of hysteresis effects, and things can come and go. But yeah, but I wouldn't worry about it in terms of like emergent pathogens. There was a paper that came out that I was asked to comment on, and I think my comment was I would be much more, because they had said that they had found viruses in ancient permafrost, and the melting was going to release these pathogens which could potentially kill humans and all these sorts of things. And my comment was, I would be much more worried about the sea level rise and the millions of people living on the ocean that are going to be underwater as a result of melting glaciers than anybody's going to die as a result of viruses being released from melting glaciers, right? I think there's much more direct effects than being exposed to new viruses. So, but good questions. Good questions. So,
and just wild thoughts, but I just wonder if those viruses are shaping the ecosystem in maybe in ice age or whatever, because in the cold days, so they were frozen in the glaciers, and today is global warming. Would it be possible just those viruses comes out and our temperature just drop down because they just reshape the ecosystem to the cold ages? Just wild guess.
They definitely shape ecosystem. We can see that. I mean, it's amazing. Like there's certain groups where the virus is completely, even, you know, like if you look at temporal dynamics in the oceans, there are certain groups of phytoplankton which are repeatedly lysed every year. They're just lysed as a result of viral infection. And so and you can see they just collapse and all these other species come up. And so they definitely shape ecosystems. And there's a fellow who is, well, if anybody has geoscience, a guy called Cesar Emiliania, who is a famous geologist, and there's a phytoplankton named after him called Emiliania huxleyi. But he was somebody that worked on foraminifera in the ocean. And I remember we published our first paper when I was an assistant prof in Texas, which the first time we did an experiment on viruses in the ocean, it worked out beautifully. We got a nice paper out. And he called me up and he said, I knew it. You know, I published this paper in the 1960s because I was looking at foraminifera in the ocean. And he looks at these profiles. And he says that, you know, we know about mass extinctions when whole groups of taxa go together. But he said with the foraminifera, it was really interesting because he kept seeing in the geological record a single taxon which would disappear globally. And it would be a single taxon. It wouldn't be a global extinction. It would be a single taxon that would disappear globally, and it would inevitably be the most abundant taxa. And he said, I said it's got to be a virus, because nothing could explain just global extinction of a single taxon that quickly. And you know, he's right. I mean, there's nothing. I mean, we're just lucky that AIDS doesn't get transmitted like influenza. There's absolutely no reason that it couldn't, right? Evolution's not very smart, right? I mean, it just, oops, I made a mistake. I killed my host off completely. Oh, well, I guess I'm dead too, right? But that's the way evolution works, right? And so there's absolutely these mass extinctions that occur. And I think that's probably, those kinds of things, processes probably happen many, many times where where a pathogens emerged, completely wiped out a whole taxon, and just created another niche for other things to go in. Yeah, so.
Okay. I think so we can continue the discussions just outside of this classroom because we have the reception. So I'm pretty sure many other people that have the questions for the Professor Suttles. So please join me to thank the Professor Suttle for the very inspiring talk.